So SWIFT-Seq is a single-cell sequencing-based blood test. Also works on the bone marrow, but really the benefit, the goal of developing this test was so that we could enable molecular profiling from the blood. So the power, I guess, of SWIFT-Seq is that it combines RNA-seq and B-cell receptor sequencing, and so in contrast to prior methods that usually rely on protein level expression of markers of malignancy that may or may not be expressed by all tumors, with SWIFT-Seq, we are going after the native barcode of the tumor cell using B-cell receptor sequencing. So the hope is that it's more accurate and it doesn't really rely on these markers that may or may not be there. In addition to detecting these cells in the first place, so circulating tumor cells in the blood, it can actually also molecularly profile them. So we are showing that it can reliably cytogenetically classify the patient. So we can infer the presence of translocations, we can infer the presence of hyperdiploidy, and these are major genetic events that characterize myeloma tumors. And on top of all of that, it can also measure the proliferative rate of the tumor, which we know from past studies is actually a powerful prognostic tool. It can also measure the expression of gene expression signatures. So it's sort of, we can think of it as an all-in-one type of test, which we think makes it very cost efficient. You know, oftentimes with single-cell sequencing, we tend to think of these assays as expensive, but in reality, they enable you to measure so many different types of information in a single test and in a single sample that otherwise you would need a bone marrow biopsy, FISH testing, gene expression profiling, flow cytometry. So taken together, as well as given the constant decrease in sequencing cost, it's actually a very cost-efficient test.
As a clinician, when I see patients with multiple myeloma or precursors in the early settings of MGUS and smoldering myeloma, we need the bone marrow biopsy, which is painful, and none of our patients want to have it, and we don't like to repeat it. And even when we do a bone marrow biopsy, we still do a very old technology called FISH, which gives us some of the information of the genetics changes that happen in myeloma, but not all of the information, meaning we get some of the translocations, the main events, but we don't get mutations. We also can get information on only the DNA, but we don't get information on the RNA. And we know from the very old data of gene expression profiling that it matters to also get information on the RNA. And we get all of that as piecemeal only in a bone marrow. So how can we overcome all of those problems so that we can enable precision diagnostics and now can help our patients get to know, are they high risk or not? Should they go on therapy or not? Which therapy should they benefit from? And by doing SWIFT-Seq, you can sort of get some of that information already. One, you can get it in the blood, which matters a lot for our patients. In fact, we have many patients already calling us saying, can I get SWIFT-Seq purely because it's a blood biopsy and not a bone marrow biopsy? Although, as Romano said, you can also do it on the bone marrow, but the idea is that you can do it on the blood. And you can do it from as few as 10 cells, which is, again, some of that innovation that if you only have 10 cells in a blood sample, you can get all of that information. Then, as Romano said, you can get all the information you get from FISH, which means I can tell the patient that they have a translocation. Again, it's a way to say that the RNA matters for the DNA, but you can say that indeed the 1114 translocation we can tell in a patient, or a 414, or something called hyperdiploid or not. Then I can get the information of the RNA, how fast those cells are cycling or proliferating, which is very critical for us to say, is this bad prognosis or not? And if you put all of that together, hopefully that could be a very strong clinical test that can matter for our patients to predict response resistance, to predict prognosis, and to help us in decision-making for our patients. I would probably say everyone, for all the way from MGUS to relapsed refractory. And the nice thing is at the very end stages when we have extramedullary disease, sometimes the bone marrow does not reflect what's happening in other sites. And the blood is actually reflective of everything that's happening in your body rather than only one piece. Remember, a bone marrow biopsy is taking a look at only one area and ignoring the rest of your body. A blood sample will actually get cells from every other place. So you may actually identify changes that are happening from another site, especially an extramedullary site that would not have been seen in a bone marrow biopsy site.
And I can chime in quickly to say, everyone and very frequently too. This is, I think, a key problem that we have currently, where because all of our molecular profiling relies on bone marrow biopsies, we can only do so many bone marrow biopsies in the patient's course of disease. And so in contrast, we very frequently pull blood samples. So this hopefully will also allow us to get the molecular footprint of the tumor much more frequently.
Well, we hope it will replace it. I think innovation and starting new technologies is always the way for us. At the beginning, maybe we will do head-to-head comparisons, but in the future, we should be using other methods compared to fish. We all like fish, but it's time for a 50-year-old technology to allow other new technologies to come along.
Yeah, I agree. And I think it's because of the additional information and the increased sensitivity, increased accuracy, and convenience too, right? We cannot do fish on a blood sample, but the assay works better. It can work off of very few cells, when with fish you typically require your bone marrow biopsy to be packed with tumor cells for it to give you reliable results and not get false negatives. And it cannot tell you what the proliferation rate is. It cannot tell you, the expression level of a progression signature. So you get a lot more for the one assay that you conduct. So I think some of the things we have to consider is, obviously not all myelomas like to. So when we're thinking about applying SWIFT-Seq on a blood sample, which I think is what most people are excited about, we need to have enough tumor cells circulating in order for the test to be able to capture those. It does work with very few tumor cells, but those still need to be circulating and be in the sample. And we know that based on sort of the biology of the tumor, some myelomas like to circulate more than others. So that is to say that, for some patients, it may just be harder to detect that. That being said, this is usually a good thing for patients, because myelomas that circulate more, usually the prognosis is worse. So even if the test can't detect a circulating tumor cell, that the news is not bad. The news is probably good, because the test is actually fairly sensitive. So I would say, as a limitation, this would be it. Trying to understand as a next step, how many patients can we actually capture using such a test? What time points? For example, after therapy, there's such a thing as minimal residual disease. We still need to understand, what the sensitivity of the test is in that setting. So there is additional work to do still.
Yeah, and I would add that maybe having the DNA information, like the mutations, again, this could improve and we can still use the same method that we're using, but having that additional information of the mutations could be very critical too. This is an academic effort. We're hoping that this will be commercialized and available clinically, so that all the patients have access to it, and we democratize what we've done in the lab. But this is hopefully coming up in the future. Yeah, we're hoping in the next few months or so, this can become a commercially available test, but we're definitely looking forward to that.